Hello. So what we're going to cover now is we're going to talk about what happens inside the cell that enables you to carry out reactions and to get energy from the food that you eat. So all the chemical reactions inside of your cell are called metabolism when you take them all together. Metabolism is one of those words that people kind of use, like we talk about somebody having a slow metabolism or a fast metabolism, but technically metabolism is all of the chemical reactions occurring inside of your cells. And we're specifically going to focus on those involved in getting energy and providing energy for your cells to function. And I'm going to talk a little more in detail about enzymes. I've mentioned enzymes, that those are proteins needed for chemical reactions. So we're going to talk a little bit more about the process by which, excuse me, enzymes work. So when you eat something like carbohydrates, say you have glucose, that glucose enters your cells. This is a muscle cell. And the energy provided in the bonds of that glucose are used by your cell to do work. In a muscle cell, that could be muscle contraction, where you're able to move your muscles and move and walk and talk, etc. We're not 100% efficient at doing that, so we also produce some heat in the process of getting energy from our food, and that's what generates our body heat, or also these metabolic reactions. So that's a bit what we're going to talk about in more detail. So again, metabolism are all the chemical reactions that occur inside of your cell, and there are thousands of them. The things that you start with are called reactants. They also are sometimes called substrates. And I think I tend to use the word substrates more. And I'll identify them as I show you specific reactions. And what you end up with are products. And generally, we do reactions, some of which are catabolic. Remember, we talked about those. Those break big things into little things. They also release energy. So that would be like breaking down the food that you eat. And then we can use that energy to do anabolic reactions where we make things. So they kind of work together. The catabolic is what releases the energy that is used by the anabolic reactions to provide materials that your cell needs. We're going to talk a bit more about the energy involved and what that is in the cell. Energy is kind of one of those hard things to understand. We talked earlier about there being two general types of reactions. Catabolic reactions are where you break things down. It's also called hydrolysis, where you use water. Lysis, remember, means to break apart. In fact, I didn't mention that, but lysosome, remember we talked about that as one of the cell organelles. Those are parts of the cell that break things apart. That's why they're called lysosomes. And catabolic release energy. The opposite type of reactions are the dehydration synthesis. They're anabolic. They're where we make things, and those require energy. So if you look at an example of that, excuse me. Here we have a larger polymer with 3 monomers. We add water for hydrolysis, so we're going to break it apart, and now we have a polymer with 2 and then 1 is free. We could break this one apart and then we would have 3 monomers. So that's hydrolysis. We're taking a bigger molecule and breaking it into smaller molecules. The opposite of that is dehydration synthesis, which is anabolic, and that's where we're taking 2 smaller. So here we have a 2 and a 1. We're going to remove water and form a 3 monomer product. So this one has 3, so we're combined a 1 and a 2 to make a 3. So that's making something bigger. So that's anabolic, and that usually requires energy. So we would need to put energy in for that reaction to occur. It would not occur spontaneously. So some examples, and we've talked about these a little bit when we went through the organic molecules. So you can combine two glucoses by dehydration, remove a water, and now the two glucoses will bond with each other. And so you have a disaccharide and water. So that's dehydration. And in this one, if we're going this way, these are the reactants or substrates, and these are the products. However, we could go the opposite way. So say you ate some maltose or something with maltose in, and you wanted to break it down into glucose. Then you would do hydrolysis. You would add the water to break that bond. You would be going this way, and you would break the maltose into two glucoses. So that would be a hydrolysis reaction. And in that case, this would be the substrate, what you're starting with. And the glucose would be your product. So depending on which way you're going, your products and substrates vary. This is just another one. This is making, when you're making proteins. You can combine amino acids. That's dehydration reaction if you're combining them to make a peptide, because they're now bonded to each other. If you ate some protein and you want to break it down to your amino acids, you can go the opposite way and break that bond by adding water back, hydrolysis, and now you're back to having two amino acids. So that's how they work together. Hydrolysis is when you break a bond with adding water. Dehydration synthesis is when you join two monomers together and take away a water. So in the cell, you produce energy, and that energy inside of the cell is used to do the cell's work, whether it's an anabolic reaction, whether it's something like muscle contraction. And so as we go through energy metabolism, we're going to use glucose as the example, as a carbohydrate. And I'll talk later about that we can get energy from fat and from protein, but it's typical you kind of use glucose as the example. So if you remember, glucose is C6H12O6. That's a molecule of glucose. So you ate something with glucose, it gets into your muscle cells, and there it gets broken down into carbon dioxide and water, which are smaller molecules. So that's hydrolysis. And that releases energy. And so that energy can be used by the muscle cell for doing muscle contraction or keeping the cell alive for a variety of different functions. So muscle contraction is a form of work that the cell does. So if you wiggle your finger, you're contracting muscles. The way our body works, our skeletal muscles are attached to bones. And so if we want to move, we contract the muscle, which means it gets shorter. And as it does that, it pulls on the bone, and that's what enables us to move. So that contraction of the muscle requires energy. And so you get the energy from breaking down glucose, supplies the energy for the muscles to contract. Excuse me. Now, when we talk about energy in the cell, that's kind of vague, and food provides energy. Like you probably have seen on labels of food, the number of calories provided by carbohydrates and proteins and lipids. And that energy in glucose, for example, is in the chemical bonds holding the molecule together. And when you break those bonds, so when you break down glucose, that releases energy. And what the cell does, it captures that energy in another compound that has a long name called adenosine triphosphate. And because it has such a long name, we tend to call it ATP. And so ATP is the form of energy that's actually used in the cell to do the cell's work, such as muscle contraction. We talked about active transport requires energy. So ATP is used by the cell to make energy. We don't store ATP, so every living cell has to constantly be making ATP. And then that ATP is used for other reactions to provide energy for those things to happen. The molecule of ATP, excuse me, if we look at it, it's a big molecule, but it's called adenosine triphosphate. So the A stands for the adenosine part, which is right here. And then the TP stands for triphosphate. And that's these three phosphate groups here. And the bonds between the phosphates is what is a high energy bond. So the energy, when you break down glucose that you ate, that's released, is used to make the high energy bonds in ATP. So what's used is ADP, which you might be able to guess is adenosine diphosphate. So it just has two phosphates, not three. This one has three, this one has two. So you take the energy from your food, and that's used to add a third phosphate. So that energy is now contained in this bond, and then that's used to do the cell's work. An example of work, and we'll talk more about what counts as cell's work. This is a lightning bug or a firefly, and so it has the ability, it has fluorescent molecules in its tail that light up, and that's an energy requiring process to light up its tail, and so it uses ATP to produce the light in its tail. They don't have lightning bugs here in Arizona. It's too hot, I guess, in the summer. I don't know if they have them up north, like around Flagstaff. I grew up on the East Coast, and in the summer, in the evening, there would be lightning bugs flickering everywhere in the neighborhood that you would be able to see. So that's just a kind of visible example of a cell using energy to do work. So when we talk about ATP, we're talking about the actual molecule inside of the cell that does the work inside of your cell. You don't store it, so you don't have oversupply of ATP. You have to constantly be making ATP. We're going to talk in the next topic about how we make the ATP. So muscle contraction, we said, was another way that the cell needs energy to contract your muscles. If you take anatomy and physiology, you'll learn this in detail. But our muscles work by two protein fibers sliding over one another, and that contraction, that shortening of the muscle fiber, requires ATP. And so anytime you're using any muscles, whether it's doing physical exercise like walking or running, but even talking or moving your hands or taking notes, you're using hundreds of molecules of ATP for your muscles to be able to contract. So some of the other type work that we do, so we just talked about mechanical work. That would be our muscles contracting. That requires ATP. Flagella, spinning, cilia beating. That would be mechanical work where we're actually moving something. Transport work. We talked about active transport. That requires ATP. Exocytosis and endocytosis, where those vesicles merge with the membrane, that requires ATP. And then chemical work are those anabolic reactions where we make things like proteins, like insulin and hemoglobin, and all the other molecules we make inside of our cells all require ATP. So anything that's anabolic, where you're making something and not breaking something down, That requires energy, and the form of energy that's used by the cell is ATP. So you use millions of molecules of ATP every day, and when you use one, say you wiggle your finger, you use up some ATP, it gets broken down to ADP, and then using the energy from your food, you convert it back to ATP. So you're constantly doing that in every one of your living cells. The way that our body works is that when we do chemical reactions, for example, we burn glucose in our cells for energy. That's the same overall reaction that if you burn a piece of paper. Paper is cellulose. Remember, cellulose is a polymer of glucose. So if you put a match to a piece of wood or a piece of paper, you're burning that paper. That's releasing energy. It's given off as heat and light. That's the same chemical reaction that happens inside of our cells when we break down glucose. We produce carbon dioxide and water and energy, but we do it in tiny controlled steps inside of our cells. We don't have like big flames going and fires and stuff. So instead of doing one big step, going from glucose, breaking it down, we do it in lots of tiny steps where we can kind of harness that energy in a better way. So that's called a metabolic pathway. So a metabolic pathway is the individual steps that are gone to get from the beginning to the end. So if we were starting with glucose and we're ending up with carbon dioxide, which is what we exhale, and water, which is what you form when you burn paper, we don't do it in one step. We do it in many tiny steps. So that's called a pathway. So to get from A to G, from the beginning to the end, we have lots of intermediate steps along the way. So we do it in a much more controlled way. And the way that we control it is with enzymes. So a couple things to remember about enzymes is enzymes are proteins. We talked about proteins. They're made from amino acids. We have inside of our body, about 75,000 different proteins, and many of those are enzymes. We have thousands of different enzymes. They're what you call a catalyst. So a catalyst is something that speeds up a reaction. So it makes it go faster. Okay. What you start with are called substrates, and then the enzyme converts them into products. It speeds up that reaction. The naming of enzymes, the official name of enzymes always ends in ASE. So you can tell something is an enzyme by its name. Sometimes there's names that aren't as familiar, but for example, lactase is an enzyme that breaks down lactose. Sucrase is an enzyme that breaks down sucrose. So they have that ASE ending indicating it's an enzyme. Okay, so if we have that metabolic pathway that we just talked about of starting with glucose and ending with carbon dioxide and water, and if we have many different steps, each one of those steps require an enzyme. So this pathway, and this is not a real pathway, it's just a made up one, of going from starting with A and ending up with G actually requires 6 different enzymes. So that's how many enzymes you would need to get from the substrate to the product. And that's how we do little control steps. And then by controlling this enzyme and how much of that enzyme we have, we can control how much of the product we're going to make. So when an enzyme is going to work, remember the substrate is like the reactant that it's going to start with. So this blob here, remember proteins have like these unique shapes. And so the enzyme has what's called an active site. And the active site is where the substrate attaches. And you can see the way they've drawn this, that it's drawn so that the active site fits perfectly. It's kind of like a piece of a puzzle, fits perfectly with the substrate. And that's true. The interaction between the enzyme and substrate is very precise. So that's why we have the enzyme lactase, which breaks down the disaccharide lactose, and then the enzyme sucrase breaks down sucrose. So they're both disaccharides, but the enzyme lactase only breaks down lactose, doesn't work on sucrose. Sucrase only breaks down sucrose, doesn't work on lactose. So they're very specific for their substrate. So the substrate attaches, the reaction occurs, and then you release the products. In the case of lactose, that's glucose and galactose. And then the enzyme itself is not destroyed by the reaction, and so it can go back and start over, grab another molecule of substrate, break it down, release the products, grab another one, and it can continue. Eventually it'll wear out, but it can break down many molecules of its substrate before it wears out. So enzymes, we tend to give examples of enzymes of breaking things down, like breaking down lactose and breaking down sucrose. So we do need enzymes to break things down. However, we also need enzymes for all the steps involved in making something. So it's not just breaking down that requires an enzyme, but it's also synthesis that requires an enzyme. So this is a little bit of a small picture. But here it's showing where we have a substrate that is bigger. It attaches to the active site of the enzyme and it gets broken down to the product. So that's a degradation where we start with bigger, ending with smaller products. But suppose you wanted to make something. You could have multiple substrates which would attach to the enzyme's active site, and they get joined together so that the product, here you're making something rather than breaking something down. Doesn't matter if you're breaking down or making something. Every reaction in the body requires an enzyme. And when we get to genetics, you'll see that many genetic disorders are due to not making an enzyme properly, and then whatever reaction that enzyme controls, you can't do properly. This is just an example of some things you may have heard of that actually contain enzymes. This is meat tenderizer. And that has an enzyme that breaks down proteins. It's extracted from papayas. And so if you had a tougher piece of meat and you sprinkled the meat tenderizer on it, the enzyme would break down those proteins a little bit so that they're not as long and tough proteins, and that would help tenderize it. The browning of curds that are turning black of looks more, but you know how like if you cut an apple, it turns brown? That's called enzymatic browning. There's an enzyme inside the apple that when it's exposed to air, it produces a brown color. If you don't, when it's not broken into, it's not exposed to air, so you don't have the reaction. And this is something to prevent gas. It also contains an enzyme. If you have lactose intolerance where you can't break down lactose sufficiently, that's due to an enzyme deficiency. And you can take lactate pills, and what those pills are actually the enzyme lactase. And so if you take that at the same time you're eating the food containing lactose, it'll help break down the lactose if you don't make enough enzyme on your own. There are some other things that will influence enzyme activity, and I'm going to talk about each of them. We're going to talk about temperature, pH, cofactors, and inhibitors. So enzymes work best at a specific pH. I realize now that we're not in the order that's on that list, but that's okay. And the pH that they work best at is usually the pH at which they normally exist. For example, these are two different enzymes that are involved in breaking down proteins in your intestinal tract. This is not their official name with the ASE. So one is pepsin and one is trypsin. And pepsin is in your stomach. And your stomach has a lot of hydrochloric acid. And so the pH of your stomach, the fluid in your stomach, is in the range of 2 to 3. So pepsin works, this is showing how much it breaks down something, works best at that pH. Trypsin is in your small intestine, and it breaks proteins down there. The pH of your small intestine is slightly basic, so trypsin works best at a pH of 8. So if you were to put trypsin in your stomach, it wouldn't work. And if you were to put pepsin in your small intestine, it wouldn't work, because they only work at a certain pH range, and that's the only time they're effective. So sometimes you might have used, like when you're making fruit salad and you're trying to keep your bananas and apples from turning brown, you can sometimes add lemon juice, and lemon juice is an acid. And so that acid inhibits the enzymes, and that will help slow down that enzymatic browning of the fruit. Temperature is also a factor. Usually the enzymes inside of our body work best at body temperature, which this is in Celsius. So 37 degrees Celsius is equal to 98.6 Fahrenheit. So our body temp enzymes work best at our body temperature. That makes sense because that's where they exist. You can slow down enzyme activity by putting it cold. So one of the reasons we refrigerate food is to slow down the enzymes that would be breaking down the food, and so that prolongs the shelf life. You put it in the freezer, even slow it down more so it lasts even longer. High temperatures generally break down the protein, and so the enzyme doesn't work anymore. So if you look, this is one of our body at a high temperature, it doesn't work. So this enzyme, after you get about 42 degrees or so, it's no longer functional. There are some this is an enzyme from a bacteria that lives in hot springs, and you can see that because it usually lives in quite hot water, its enzymes work best at about 70 degrees. Our enzymes would be totally non-function. When an enzyme breaks down, that's called denaturation. And heat does that. Like the white of an egg is mostly proteins. It's not really an enzyme, but when you heat the egg, it turns like from that kind of opaque color to the solid white, and that's due to denaturation of the proteins. So that if that protein were an enzyme, it wouldn't work anymore if it was denatured. Some enzymes also require cofactors, which is another molecule that's needed. So this big blue blob here is the enzyme. This is the active site, and sometimes cofactors are needed for the substrate to be able to attach properly to the enzyme. So that little red thing here is the cofactor. So if the cofactor wasn't there, the substrate couldn't attach and the enzyme wouldn't work properly. An example of that, I thought I had a slide on it, which is not here, so that's why I went forward, is vitamin C often functions as a coenzyme. It's needed for the making collagen, which is the major kind of support protein in your gums and skin. And so if you have a severe vitamin C deficiency, you have your skin and your gums get deteriorate, and you can get like severe bleeding of the gums and little pinpoint hemorrhages on your skin. And that's only with a severe vitamin C deficiency, which is pretty rare. But that's because the vitamin C is a cofactor for an enzyme that makes that protein collagen. You can also inhibit an enzyme by other means besides the cofactor, heat, and pH. And there are substances that can inhibit an enzyme. And there are actually three different types of inhibitors. There's competitive inhibitor and non-competitive, which I'm going to talk about first, and then feedback inhibition. I'm going to go to the pictures. We're running over in time. I'm not sure if I should have broken this up. So a competitive inhibitor is something that kind of is like the substrate, but it can attach to the enzyme, and that prevents the real substrate from attaching. Some pharmacological agents are competitive inhibitors, and so if they want to stop like a particular reaction from occurring, like inflammation, if they put a competitive inhibitor, that would block that enzyme from working. A non-competitive inhibitor doesn't block the substrate, but instead it attaches to the protein and changes its shape. So now the substrate doesn't fit right. So they both work by decreasing the activity of the enzyme by preventing the substrate from properly binding. The more common type of inhibition that our body uses is called feedback inhibition. And that's where we have this metabolic pathway that we need to make a certain product. But suppose we have enough of that product. We don't want to keep making it if we don't need it anymore. So feedback inhibition is where the product goes back and inhibits the enzyme. And it's usually the first enzyme. So like suppose you needed to make the hormone insulin and to be able to release it to lower your blood glucose. But once your glucose is normal, you don't need to keep making that hormone anymore. So you don't want to kind of waste your resources making something you don't need. So that's where the feedback inhibition, and that's where the product inhibits the enzyme. So once you have enough of something, you're not going to keep making it if you don't really need it anymore. This is just some examples. Penicillin actually works by inhibiting an enzyme in bacteria that is needed to make their cell wall. And since they can't make their cell wall, then the bacteria can't live. And so the way that penicillin works is actually by inhibiting that enzyme. The non-steroid anti-inflammatory drugs, things like Tylenol, Motrin, and aspirin, also inhibit a number of enzymes involved in inflammation and pain, and that's how they work as enzyme inhibitors. So that can sometimes be exploited pharmacologically. What we're going to talk about in our next PowerPoint is the metabolic pathways involved in getting energy from food such as glucose. And this is the overall pathway for cell respiration, which is what that's called. Our next PowerPoint is going to talk about that. And if you notice, cell respiration is the opposite of photosynthesis. So the plants are making glucose out of carbon dioxide and water, and we're burning glucose and producing carbon dioxide and water. So they're the exact opposites. So I will stop there, and I think this is the last PowerPoint for Module 2.